Your doctor handed you a prescription. Maybe it was metformin. Maybe it was something newer with a name you couldn't pronounce. Either way, the message was the same. Take this every day for the rest of your life, or your blood sugar will destroy you. What he didn't mention is that a leaf growing wild on 6 continents has been doing the same job for 4,000 years. Without the side effects, without the prescription, without the monthly copay. In ancient China, doctors prescribed it to emperors. In Korea, grandmothers brewed it into tea after every meal. Modern researchers in Japan ran clinical trials and watched blood sugar drop 30% within an hour of consuming it. Real numbers, real patients, real published studies. And then the research quietly stopped. Funding dried up. Doctors stopped mentioning it. Pharmaceutical companies developed a synthetic version, patented it, and sold it for 40 times the cost of the actual leaf. The leaf still grows wild. It still works. The only thing missing is the awareness. Today you're going to learn what your doctor was never trained to tell you. The tree has been cultivated for over 5,000 years. Not for its medicine, for its silk. Morus alba, the white mulberry. Its leaves are the sole food source of the silkworm, Bombyx mori. Chinese civilization built an entire trade route on this relationship. The Silk Road, the longest commercial highway in ancient history, stretching from Chang'an to Constantinople, existed because of this tree. Emperors guarded its secrets under penalty of death. Wars were fought over it. Economies rose and fell on it. But while the rest of the world chased the silk, Chinese physicians were quietly studying the leaf itself. In traditional Chinese medicine, the mulberry leaf is called sang yi. It is classified among the herbs that clear external heat and cool the blood. It appears in the Shennong Bencao Jing, one of the oldest pharmacological texts in existence. It was prescribed for fever, cough, inflamed eyes, and a condition that Chinese doctors described as wasting thirst, excessive urination, and unquenchable thirst. A description that modern endocrinologists would immediately recognize as diabetes. The Chinese diagnosed it, named it, and prescribed a treatment for it, over 2,000 years before insulin was discovered. Here is what the laboratories found when they finally looked inside. Mulberry leaves contain over 200 identified bioactive compounds, flavonoids, polyphenols, polysaccharides, alkaloids, amino acids, vitamins, trace minerals. The leaf is not a single compound medicine. It is a biochemical pharmacy growing on a branch. But the compound that changed everything was isolated in 1976 by Japanese researchers. They found it in the roots first, then in the leaves. They called it 1-deoxynigeromycin, DNJ for short. DNJ is an immunosugar, a sugar molecule in which the oxygen atom has been replaced by a nitrogen atom. That single atomic substitution gives it a shape almost identical to glucose, so identical that it fits perfectly into the active site of an enzyme called alpha-glucosidase, the enzyme responsible for the final step of carbohydrate digestion in your small intestine. Picture a lock and a key. Alpha-glucosidase is the lock. Glucose is the key. DNJ slides into the lock first and jams it. While DNJ occupies the enzyme, the carbohydrates you just ate cannot be broken down into glucose. They pass through undigested. Your blood sugar does not spike. That is not folk medicine. That is competitive enzyme inhibition. The same mechanism used by the prescription drugs acarbose and miglitol, which generate over $2 billion in annual revenue worldwide. The difference is that acarbose and miglitol were synthesized in laboratories. DNJ was growing on a tree. The studies are not ambiguous. They are not preliminary. They are published in peer-reviewed journals, conducted at accredited universities, and they show results that pharmaceutical companies would build billion-dollar marketing campaigns around, if the compound were patentable. A randomized, double-blind, placebo-controlled trial at Oxford Brookes University gave 38 healthy participants 75 grams of sucrose with mulberry leaf extract, or 75 grams of sucrose alone. The mulberry group showed a 42% reduction in peak postprandial glucose and a 41% reduction in postprandial insulin. The effect was immediate. It lasted approximately 60 minutes. There were no adverse effects. A separate study at Tianjin University of Traditional Chinese Medicine tested mulberry leaf extract against 4 common dietary carbohydrates. The glycemic index reductions were 53% for maltose, 34% for sucrose, and 31% for maltodextrin. The extract was well tolerated. No participant reported significant side effects. A 2020 randomized controlled trial in Thailand gave obese pre-diabetic participants mulberry leaf powder standardized to 12 milligrams of DNJ daily for 12 weeks. Fasting blood glucose dropped. HbA1c improved. Insulin resistance trended downward. The researchers concluded that mulberry leaves showed favorable effects on glycemic and insulin resistance profiles. And then you have to ask the question, if the data is this consistent, this reproducible, this well-documented, why is the leaf not on every endocrinologist's desk in America? The answer is in the chemistry and the patent office. In the 1960s, pharmaceutical researchers discovered that aminosugars like DNJ could inhibit the enzymes that digest carbohydrates. They recognized the therapeutic potential immediately. But DNJ is a naturally occurring molecule. It cannot be patented, so they modified it. They took the core structure of DNJ, altered the molecular configuration, and created synthetic analogs. Acarbose, derived from bacterial fermentation, was approved by the FDA in 1995. Miglitol, a synthetic derivative of deoxynitromycin itself, was approved in 1996. Both drugs work by the same mechanism as the mulberry leaf, alpha-glucosidase inhibition. Both drugs reduce postprandial blood sugar. Both drugs are prescribed to type 2 diabetic patients worldwide. Acarbose costs between $30 and $200 per month, depending on dosage and insurance. Miglitol costs roughly the same. Both produce well-documented gastrointestinal side effects: flatulence, bloating, diarrhea, and abdominal discomfort. These side effects occur because synthetic alpha-glucosidase inhibitors remain in the gut alongside the undigested carbohydrates, and intestinal bacteria ferment both. Mulberry leaf DNJ does something different. After inhibiting the enzyme, it dissociates from the active site and is absorbed through the gut wall into the bloodstream. It is then rapidly excreted through the kidneys, unmetabolized. It does not remain in the gut. It does not feed bacterial fermentation. The gastrointestinal side effects that plague acarbose and miglitol are largely absent. The natural version works by the same mechanism. It produces fewer side effects. It costs less than $10 a pound as dried leaf tea, and no pharmaceutical company can patent it. That is why your doctor has never mentioned it. But the story does not end with dried leaf tea. In China, researchers went further. They extracted and concentrated the full alkaloid profile of the mulberry twig. Not just DNJ, but a complex of 7 aminosugar alkaloids, and created a standardized extract called SZA. The Chinese Academy of Medical Sciences developed it. Phase 2 and Phase 3 clinical trials were conducted. The results? SZA reduced glycated hemoglobin with fewer gastrointestinal side effects than acarbose. The drug performed comparably to the pharmaceutical standard while producing less diarrhea, less bloating, and less intestinal discomfort. The researchers attributed this to the synergistic action of the multiple alkaloids working together, as opposed to the single compound approach of acarbose. SZA is being developed as a novel hypoglycemic drug in China for the Chinese market. The Western pharmaceutical industry has shown no interest in licensing it because the active compounds are derived from a tree that grows on every continent except Antarctica. You cannot build a monopoly on a plant that grows in every backyard in the temperate world. Walk into a pharmacy in Seoul, South Korea, and you will find mulberry leaf tea on the same shelf as over-the-counter medications. It is not marketed as a novelty. It is marketed as a postprandial glucose management product. Korean grandmothers have brewed it after meals for generations. In Japan, mulberry leaf tea is sold as a functional food under the country's Food for Specified Health Uses classification. It has been approved for claims related to blood sugar regulation. Japanese consumers drink it daily. Clinical trials supporting the approval were conducted at Japanese universities and published in peer-reviewed nutrition journals. In Okinawa, one of the world's 5 blue zones, regions where people live measurably longer than the global average, mulberry leaf tea has been part of the daily diet for centuries. In traditional Okinawan cuisine, it is called kuwacha. It is served after rice-heavy meals. The connection between mulberry tea consumption and the remarkably low diabetes rates in Okinawan elders has been noted by epidemiologists, though not formally studied in isolation. Across East Asia, the leaf was never forgotten. It was never buried. It was never classified as alternative medicine. It is simply medicine. Integrated into daily life, sold in pharmacies, consumed by millions, and supported by the same clinical data that Western regulatory agencies have chosen to ignore. But here is what makes the mulberry leaf more than a glucose story. The 200-plus bioactive compounds do not stop at alpha-glucosidase inhibition. Mulberry leaf flavonoids, including quercetin, rutin, and camphorol, have demonstrated antioxidant activity that protects pancreatic beta cells from oxidative damage. Beta cells produce insulin. When they are damaged by chronic oxidative stress, insulin production declines. Protecting them is not treating diabetes, it is preventing its progression. Mulberry leaf polysaccharides have shown the ability to enhance glucose uptake in muscle and fat cells by increasing the activity of insulin signaling pathways. In animal models, mulberry leaf supplementation improved insulin sensitivity, reduced triglycerides, and lowered LDL cholesterol. A 2022 comprehensive review, published in Pharmacological Research, identified multiple mechanisms by which mulberry leaf compounds influence metabolic disease. Modulation of gut microbiota, regulation of lipid metabolism, suppression of hepatic gluconeogenesis, and activation of brown adipose tissue, the fat-burning tissue that helps regulate body weight and metabolic rate. The leaf does not do one thing. It does the same thing your doctor's prescription tries to do, from 6 directions simultaneously. And it has been doing it for 5,000 years. Follow the money. The global diabetes drug market was valued at $71 billion in 2021. It is projected to exceed $150 billion by 2030. Alpha-glucosidase inhibitors alone generate over $2 billion annually. Metformin, the most prescribed diabetes drug on Earth, generates $3 to $5 billion per year, depending on formulation. A dried mulberry leaf costs less than $15 per pound. A pound of leaf makes approximately 100 cups of tea. That is less than 15 cents per dose. The Oxford Brooks clinical trial used a standardized extract dose equivalent to approximately 250 milligrams of leaf extract, a quantity that costs fractions of a cent to produce from raw leaf material. The pharmaceutical industry does not suppress the mulberry leaf through conspiracy. It suppresses it through indifference. There is no incentive to fund large-scale clinical trials on a compound that cannot be exclusively owned. Without large-scale trials, the FDA will not approve therapeutic claims. Without approved claims, no American doctor will prescribe it. Without a prescription, the public is taught that the absence of a recommendation means the absence of evidence. But the evidence is there, published, peer-reviewed, reproducible, and available to anyone who cares to look. Mulberry trees grow on every continent except Antarctica. In the United States, they are classified as an invasive species in many states. They grow wild in parks, along highways, in abandoned lots, and in suburban backyards from coast to coast. The white mulberry, Morus alba, was introduced to colonial America in a failed attempt to establish a silk industry. The trees survived. The silk industry did not. Dried mulberry leaf tea is available at Asian grocery stores, health food shops, and online retailers. A pound costs between $10 and $20. Brew it exactly as you would green tea. Steep the dried leaves in hot water for 3 to 5 minutes. Drink it before or with a carbohydrate-heavy meal. This is the timing most consistent with the clinical data showing postprandial glucose reduction. For concentrated effect, mulberry leaf extract capsules standardized to DNJ content are available at health supplement retailers. Look for products standardized to at least 1% DNJ. The dose most commonly studied is 250 milligrams of extract taken before meals. One caution matters. Mulberry leaf is not a replacement for prescribed diabetes medication. It is a complementary approach supported by clinical data. If you are currently taking metformin, acarbose, or insulin, do not discontinue or modify your medication without consulting your physician. The leaf works, but it works alongside medical care, not instead of it. The pattern is the same one you have seen with every plant in this series. A natural compound with documented therapeutic activity is sidelined because it cannot generate proprietary revenue. The structure of modern pharmaceutical development requires exclusivity. A leaf that grows on 6 continents and produces its own alpha-glucosidase inhibitor for free is the opposite of exclusivity. The pharmaceutical industry identified the mechanism. They confirmed it worked. They synthesized a version they could patent. They sold it for 40 times the cost of the leaf. And they never told the patient that the original version was growing in the park across the street. Acarbose is mulberry. Miglitol is mulberry. The $2 billion alpha-glucosidase inhibitor market is a synthetic echo of a leaf that Chinese physicians prescribed to emperors before Rome was built. The leaf still grows. The data still holds. The only thing that changed was the price tag and the patent number. The story of the mulberry leaf is not about a tree. It is about what happens when medicine cannot be monetized. For 5,000 years, this tree fed the silkworms that connected civilizations. For 2,000 years, its leaf treated the condition modern medicine calls type 2 diabetes. For 50 years, its active compound has been studied, confirmed, synthesized, patented, and sold back to the public at a markup that would make the ancient Chinese pharmacists weep. The tree does not care about patents. It does not care about FDA approval. It does not care about clinical trial funding structures. It grows. It produces leaves. The leaves contain a compound that inhibits the enzyme that spikes your blood sugar. And it does this every spring, in every temperate climate on Earth, for free. Your doctor was never trained to mention it. Your pharmacist was never taught to stock it. Your insurance company has no billing code for it. But the tree outside your window has been offering it for 5,000 years. The prescription was never missing. It was growing. A leaf that drops blood sugar in clinical trials. A compound that works by the same mechanism as a $2 billion drug class. A tree that has been cultivated for 5,000 years across 6 continents. And a medical system that synthesized the molecule, patented the copy, and forgot to mention the original. But now you know. If this vault opens something for you, subscribe to Nature Unearthed and hit the bell. Every like and every share preserves what they tried to erase. The next vault opens soon.